In the business of biotech, adaptability is requisite because sometimes, in fact, oftentimes, you have to pivot. Things like an unfavorable readout, a lost investor, a shortening runway, a pandemic, or an abysmal capital market are likely to force a pivot. No wonder that so many biotech builders pivot begrudgingly, only when they have no other choice, their hand perhaps forced by a board. Better, in my opinion, to be a proactive and optimistic pivoter, one who embraces the pivot and who pivots not in the direction of the path of least resistance, but in the direction of the path of most opportunity. I'm Matt Pillar.
And I'm Anna Rose Welch.
And on this, yet another RNA-centric episode of the Business of Biotech, we're talking with a Renaissance man who's been busy perfecting the pivot. Dr. Erik Digman Wiklund is CEO at Circio, a circular RNA developer that's the product of a recent pivot that we'll learn about today, but we'll also learn about some of Erik's preceding pivots and those he sees coming next as RNA and the circular variety of RNA continue to make waves in the space. Anna Rose, I don't know if you counted all the times that I said pivot in my life, I don't think I've said it as many times as I have.
I'm not going to lie, there were moments there that felt almost Shakespearean. You know, the cadence of your words were very poetic.
Let's pivot to Dr. Digman Wiklund. Welcome to the show. Erik, it's a pleasure to have you here.
Thank you very much. It's great to be with you.
It's great to have you. And, as you know, I kind of like to start these conversations off with sort of some background and for context on the perspectives of my guests. So I want to rewind a little bit and start with you. It's not uncommon for me to interview biotech builders who began their careers at McKinsey. In fact, I just a couple of weeks ago, I was at a conference having a conversation with Udit Batra, who's the CEO at Waters, former CEO at Millipore Sigma. He started at McKinsey. There are probably I could probably count like a dozen guests that I've had on the podcast who started their careers at McKinsey. And it's interesting to hear about like how that early perspective kind of shaped the worldview and the next steps for the biotech builders that have started there. So tell us about that experience from your perspective.
I think McKinsey or the McKinsey training that you get is particularly suited for the biotech industry. And that's because when you're at McKinsey, you're sort of forced to work on a lot of different topics, often for a short period of time, and you jump around. So whereas as a scientist, I'm a trained molecular biologist, there you go really deep on something. You need to get all the details right. And once you step out of that and go into management consulting in a place like McKinsey, it's the opposite in many ways. You need to quickly figure something out, and you need to move from one topic to the next. So I think you learn to structure and analyze information in a quicker way and get up to speed more quickly. And you need to be willing to do some hands-on work yourself. I think that jives very well with being a biotech executive and CEO, because one, you need to be strategic. You need to understand a lot of different areas. You need to understand everything from manufacturing, regulatory, preclinical development, clinical trial design. You're involved in absolutely everything. Mm-hmm. And you also need to roll up your sleeves and do a lot of the actual hands-on work yourself. And that's probably more lost in a larger corporation. You probably stick more to your department, and when you're at a strategic level, you may no longer actually do so much hands-on work yourself. So maybe that's why a lot of McKinsey people like the biotech world, because it gives you this opportunity to be either involved in everything, wear many hats.
Yeah. I mentioned from the outset that you're a Renaissance man. And it occurs to me that your, I guess, proclivity to explore expanded horizons was probably built into you prior to your time at McKinsey, because when I look at your resume, so to speak, right, like your LinkedIn profile, you left McKinsey to lead post-acquisition integration of Algeta into Bayer, and it seems like, okay, well, this guy is solidly on track, like to be a deal maker in biotech. But your next position after that was in product development with a krill-based nutrition supplement company. You did that while simultaneously founding and launching a dinner kit cooking concept.
Which is amazing.
That's just awesome. All the while running a successful, of all things, photography business. So it begs the question, like anyone looking at your CV is going to go like, well, what was this? What was this departure on Dr. Digman Wiklund's trajectory?
And maybe you can tell that I have a lot of different interests and I like doing different things. And that's probably why I didn't stay in academia and in science and went into industry. I went to Algeta, as you said, after McKinsey, and my role there was initially in strategic planning of manufacturing for the future. And then quickly the Bayer acquisition came up. And I'd spent significant time at Bayer during my consulting years, so I knew that organization well. So I think that's why I was selected to do this integration project between the two organizations. Now, I also learned from my time as a consultant that I'm much more of an entrepreneurial person than a big corporation person. So I also kind of knew that I didn't want to stick around there. So I could say I integrated myself out of the resulting merged entity in Norway. But overall, that merger was very successful, and to this day, Bayer retains a substantial, I think 2/3 of the team in Algeta. They've put their radiopharma research hub in Norway. So we were able to make sure that a lot of the value was retained in Norway. The manufacturing is still retained in Norway. But I then went on to other things, as you said. And my plan actually was to not have a boss again. So I decided, look, I'll try and start some projects on my own. I can work when I feel like it instead of having a boss. And I had 6 months garden leave and salary anyway from Bayer. So then I started a few things off. I'd always been a keen photographer and worked on that. So I professionalized this. So that was a way to earn some revenue fast, because I could take sort of wedding photography jobs and these kinds of things. I do that with my wife, so it's a fun thing we do together. Yeah. Then I started this dinner kit concept simply because actually the selection in grocery stores in Norway is terrible. Wow. So we wanted to make a food concept suitable for people who want to eat good food in busy lifestyles with kids. So we started that with 4 of my friends, and that's still a thriving business. And I'm no longer operationally involved, but I chair the board and help out with fundraising, and the company is doing well. Wow. And I set up also a consulting business to do projects, and that consulting business is what led me into the krill arena. Some former McKinsey colleagues worked in an omega-3 company in Norway. So in Norway, pharma and biotech is a really tiny industry. Energy, seafood, these are the large industries in Norway. So I also thought maybe it makes sense to position myself a bit more towards seafood and nutrition, simply because it's a bigger market, more jobs available in Norway. So I started off taking some projects, and that eventually evolved into a full-time role with a krill company. Yeah. Actually, trying to make protein powder, like for sports nutrition, from krill.
Wow.
That was the project we're working on.
Yeah. No, that's really cool. What was the catalyst for your return to biotech?
So a bunch of people who had been at Algeta sort of got scattered around, and including the CFO, who I worked closely with at Algeta. And he then later became the CEO of the predecessor company to Circio, which is called Targovax, which is an immunotherapy company. So he took over that company in 2016. And in 2017, he was looking for a CFO and contacted me. So then basically that was too difficult to turn down. Some of my colleagues, and at that time, immunotherapy was super hot. We were developing an oncolytic virus, which was also in vogue. And I think even though I've been doing a bunch of different things, I've always, since I was research scientist. I kind of envisaged that at one point in my career, I'd want to be a biotech CEO. Seemed like a really fun thing to do. And I've kind of kept that in mind, sort of make career decisions that could move me in that direction. So when a CFO role came around, I thought that was a great opportunity to learn some more of the finance ropes. Plus, I knew the CEO well. He had been the CFO of Algeta, but a non-scientist. So we had this kind of symbiosis where we could learn from each other. I could. He was a mentor in terms of on the finance and fundraising aspects, and I could be supportive on the science side for him as a discussion partner.
Yeah.
So that's how I ended up in coming back to biotech and oncology in the end. After all, even though it was I purposely tried to get out of it for a while.
All the way to krill.
Yeah.
You went all the way to the oceans.
So you came back, and the ride, the roller coaster ride, was far from over. You came back to Targovax. Tell us a little bit about how Targovax evolved into Circio.
So when I joined Targovax, the company was the result of a merger between a Norwegian cancer vaccine company and a company in Finland developing an oncolytic virus. So these companies were put together to create a bit more critical mass. They fundraised, got off to a pretty good start, and ran an exploratory trial, phase 1/2 trial program, that was starting when I joined. And so the first part of the time at Targovax was to execute these clinical studies, get the readouts, and then move the company on. Now, I think what happened is that both cancer vaccines and oncolytic virus, which is what we were developing, went a bit out of favor. Cancer vaccines are maybe having a bit of a renaissance now.
Yeah.
But unfortunately, several clinical failures made it very difficult to finance. So it's difficult to excite investors, even though we had good data. And then in the middle of the development, we were focusing that cancer vaccine. It's a vaccine targeting KRAS mutations in the resected pancreatic cancer setting. And right in the middle, when we were planning our phase 2 program, the standard of care changed, dramatically extending the expected survival. So we were then looking at having to do a survival trial where expected median survival would be between 4 and 5 years. Yeah. And that was just impossible to fundraise for. So we would just put our vaccine on top of whatever was the standard of care, but you can't get an investor to put money into such a program where it's going to take 6-7 years before you have a definitive readout. So that kind of pulled the rug under that program a little, and we had to step back. And as we did that, sort of realized we need to pick a new indication and restart phase 1 in many ways. We also decided to upgrade the product itself. So we took 2 years to modify both the formulation, the adjuvant we used, as well as find other combination strategies, and then put it back into patients last year. So now that program is actually returned, and I think cancer vaccines are a bit more in favor these days. So that's been resurrected. The other program, the oncolytic virus program, that was really when we started a field that was very much in vogue. People were quite excited about oncolytic viruses, but then slowly there were readouts that were negative. TVEC from Amgen hasn't performed commercially as well as was anticipated, and people became really skeptical. So I think what happens is the burden of evidence required to move on or get fundraising or a partner changes or becomes more substantial. So when the program was started, this was hot stuff. So people, let's just do a quick trial, 20 patients, get a signal, then we can partner out the program. That was the thinking. But when that work was done, people wanted to see 100 patient data, preferably randomized data, to be able to make a decision, because a small trial simply didn't cut it. So even though we had strong data, I would say, it wasn't a large enough data set or robust enough that we could get sufficient interest to pursue it. And then unfortunately, we just had to pull the plug on that program.
Yeah.
So where did circular RNA come in? How did you end up in this gene therapy world?
Yeah, so the 3 years ago, the company was in this situation where it was becoming clear that it was difficult to pursue the oncolytic virus program. Mm-hmm. The next step had a price tag of around $50 million. The trial that needed to be done and associated CMC work, we were able to fundraise maybe a third or half of that. But then we decided, look, it's doing another sort of half-baked, not properly sized program. We know it's going to be hard to partner this out. It's only worthwhile if you really do it.
You can give it your all, right? If you can actually give it your all. Yeah.
We could have done sort of another 40-patient single-arm trial, gotten some signals, but I don't think it would have moved the needle. Mm-hmm. Yeah. So that was one aspect. And also at that time, our cancer vaccine was not in any active trials. So the board was discussing, is it better to throw in the towel, what to do now? We still had some funding left. And at that time, I of course, I worked on circular RNA back when I was a researcher and published some of the very early circular RNA papers together with a very talented colleague of mine, Thomas Hansen. And we published an initial paper in 2011, and after that it became this big thing. So this evolved into big fields. So I sort of tracked over the years, and then in 2019 it was shown that you could use circular RNA to make express proteins in vivo. And then in 2021, which is just a few months before we were in this situation, Orna had launched. Yeah. With their synthetic circular RNA program. Laron followed closely thereafter with a massive financing round. So suddenly, circular RNA was this ultra hot thing. Yeah, which I then naturally wanted to get into. Yeah. So I pitched this to the board. I said, look, I know circular RNA. I can recruit Thomas, who is our sort of founding scientist of this field and is very knowledgeable. And we can build a circular RNA company instead of pursuing these legacy immunotherapy assets. Sure. And the initial thinking was that we could use the viral vector we had. So for the oncolytic virus program is based on an adenovirus. So we had substantial adenovirus knowledge in-house. We had access to a clinically validated vector. And the initial idea was to make that adenovirus express circular RNA. And circular RNA has the advantage of being more durable. It'll hang around in the cell for longer, and it makes more proteins. So we thought this would be an interesting tool to use to make a virus for cancer that can express a therapeutic payload at higher levels for longer. Yeah. And I had two conditions to take the job in 2021, and that was one, that we move in this circular RNA direction, and two, that I could recruit Thomas to the company, because it would be impossible without having someone with deep scientific knowledge. Yeah. So,
you wanted to return to your roots. You really needed to.
So rather than a pivot, it was maybe a circle coming later.
You resurrected a former interest, yeah.
Fantastic. Now I have to go back and re-record the end.
I know, you can't use Renaissance Man anymore.
Stop saying pivots so much. The circular, it occurred to me as I was preparing for this conversation that last week we did an episode. I think it was the second one that Anna Rose joined me on, that was RNA focused with Radar Therapeutics. And I didn't give you, like, for listeners who are like maybe missed the first time you recorded with me, I never gave them an opportunity to like understand why you're here. So I'm going to put you on the spot.
I just decided to jump into the room.
We made
randomly.
I think on that first episode, I think I made it clear, like, from now on, whenever I'm interviewing someone from an RNA company, I'm going to have
I'm an interloper.
An RNA interloper. Anna Rose is she's the community director and chief. What is your title?
That's an excellent question.
She runs.
I'm the editorial and community director for Advancing RNA, which is one of the newest Life Science Connect publications. So we focus on all things RNA. Predominantly, I focus on the encoding modalities, so your linear self-amplifying and your circular RNA. But we also do cater to and are always looking for content on the non-coding side of the industry as well.
There you go. So I made the decision that anytime I had an opportunity to speak with someone from the RNA world, Anna Rose was going to join me. And then
I think it kind of forced his hand.
Yeah, Anna Rose made,
too.
unbeknownst to me, she made the decision she was going to start feeding me RNA companies so that she could take over the business of biotech.
So if any RNA companies are listening, please pitch us. I will fight for you.
Yeah. So now I let Anna Rose come in and ask questions specific to the RNA space.